Hello everyone, how are we doing today? So today we're going to be going over this little laboratory model video, going over the digestive system. So we're going to go over the major organs and major parts of the digestive system, going all the way through the system. And then we're also going to look at microscope image of the small intestine. We could look at so many histology samples from the digestive system, but in this little video today, I just wanted to show you an example from the small intestine. It's a good way to highlight the different layers we find in this tube network that runs through us. So this is just a video I'm going to talk through today. Now, I'm not going to draw lines and write specifically on it like I have on other videos. Just too much to label here to make the video too long. So I'm going to talk over it as we go through here. So I take the different parts apart and we look at the different regions in here. Then we look at, you're just looking at the large and small intestines, you're going through that part, and then we jump over and we look at the liver as well. So just playing through the video one time here. So there's the liver, looking at this is the anterior aspect of the liver, and then I flip it over and then we look at the posterior aspect of the liver as well. Both directions are important, so we'll name the structures in there. And then we also have another model in the laboratory. Is this little poster thing, and this shows the structure slightly differently too. And I like some of the things on this, such as the villus right there, showing the lacteal and stuff, showing more of the gross anatomy of the entrances to the tube and some different layers of the stomach as well. So that's the essence of this video. So we're going to go through here and starting at the oral region. So to do that, I want to start right over here. So when you first eat something, you form that food bolus in the mouth, but then you also have some important glands here. This one is the parotid gland, and this one is the submandibular gland. There's also a sublingual gland under here, but these are two of the bigger ones. So all of these are salivary glands. They release saliva into this region to help that food get mixed with your saliva, and it creates that bolus. Then it comes back here. So this is the oropharynx, the back of the mouth. Then it becomes the laryngopharynx, and then the esophagus comes down through here. The esophagus goes all the way to the stomach then. So these are just a few important parts of the mouth and accessory organs of the mouth. So as we continue through here, we then go down into the stomach. So I can start out with showing this one, and then I can go over and show the one from the actual model. So the bolus comes into the stomach here at the cardiac sphincter. Now the stomach has a lesser curvature and a greater curvature, so just two different length curvatures. The top part of the stomach is called the fundus. Now it's hard to see on this model here, but it is highlighting these three layers right here. So the stomach is unique in the fact that it has three different orientations of its smooth muscle around it that acts as so it can do the peristaltic wave and also have a very good grinding mechanism. Inside the stomach then are these little ridges. These are called the rugae. This is the pyloric canal down here. So the food bolus, or mixed with hydrochloric acid and all that good stuff, pile up down here, and then they're slowly put into the duodenum, which is the first part of the small intestine right here. And this sphincter right here is called the pyloric sphincter or pyloric valve. So that pushes the food in, and then in the lecture we talked about all the feedback mechanisms of how just having the chyme, it's now considered chyme, in this region can impact the stomach. So all these different feedback mechanisms that take place. Now I do want to go back to the model and show how this is oriented here. So here, looking at where the liver is over this whole region, just doing a quick panorama down through here. I then remove the liver from the model. So here, when we're looking at this, right here would be the esophagus coming down through, and then right here would be the cardiac sphincter and pyloric sphincter. Now, on the stomach model, as I play through this now, I zoom in to the three different muscular lengths here. So there's a horizontal, oblique, and transverse right there. And then this is looking deeper into the stomach, where we can see those ridges in those rugae that form, and then down here we see that pyloric sphincter. So then I remove the stomach, and look what appears. Right beneath the stomach we have the pancreas. So this little structure right here is the pancreas. I think it goes down right there. And then this little tube here is the pancreatic duct. The bile duct connects with that, forming the ampulla that then enters into the duodenum. This is an accessory duct of the pancreas. So the pancreas is producing a bunch of the hormones and enzymes required for digestion. So very, very important products are made there. Talked about those in the lecture. Then over here, since I didn't mention this for the lymphatic system or show this model, this is the spleen. So right over here is the spleen, which is a really important part of the lymphatic system. But I talked about that then. It just could be something I'd label on the practical for this one. All right, so let's clear that and continue through this video. So there's the pancreas. The pancreas is also shown on that other model, but it's not that good. And right there is the kidney. I didn't label what was back there. So kidneys are back there. And then we get to the intestine. So down here, I'm looking at the different portions of the intestine. So this is the small intestine through here. Small intestine is broken up into 3 regions. The first 10 inches or so are the duodenum. Then we have the jejunum, and then the last portion is the ileum. So if I were to label it, jejunum would be over here, and ileum would be anything that's towards the distal portion of the small intestine. Now, where the ileum enters into the large intestine, it enters through the ileocecal valve. And I zoom in on that valve a little bit right here. So that valve is that little spot right there, and that's where the ileum connects to the cecum. So this region of the large intestine is known as the cecum then, and then it goes up, the up portion. And then down there you also see the appendix hiding. But I take the intestines out and show a better representation of them coming up here anyway. So here I remove them, and we'll look at them. But here I zoomed in here to show, that's how the duodenum then wraps around there. Then we go over here and we look at the intestines a little better. So here I then zoom in. We can see the appendix down there now. There's the ileocecal valve. And then I also check out the sigmoid colon. So let's look at the different parts of the large intestine. Now, so let's find the angle here. So right here would be the cecum. And then I didn't get a good angle of it, but the large intestine goes up first, or ascends. That's called the ascending colon. So if I were to label that, it would be anything above this point right here on the practical. And then the part that goes across is the transverse colon. The large intestines are unique, and they have like these little pockets. These are called haustra. And then there's also a little ligament of muscle and stuff that go through here called the tenia colia. And then there are also some little fat pockets that aren't shown too well on this model, but those are called epiploic appendages. So then this is a transverse colon. Colon then comes down and is the descending colon. So very nice naming structure here: ascending, transverse, descending. But then underneath the colon does a little wrapping mechanisms. It forms like a little S down here. So I get an angle here. You can see this little S structure. This is called the sigmoid colon. And then it empties out through the rectum here. And then right here we have the anal canal. So if I were to label this region, right here would be rectum, right here would be anal canal, but either would be fine. Because, practically there's no like clear line indicating the difference. So then there's transverse colon and so forth. I then flip it over, but there's nothing really, there's that tenia coli you can see on the back, but nothing too extra back there. So then we move on to the liver here. So the liver, we have 2 main lobes we can see on this side right here. The larger lobe is the right lobe, and this one is the left. So this is the anterior aspect. This little green spot down here is then the gallbladder. So now I do flip it over to look at the posterior aspect, and right there's the diaphragm on top as well. So then here is another important aspect that I want to draw on this one. So now the right lobe is over here. Again, this is posterior. Here is the left lobe. Now there are 2 other lobes we need to talk about. Here is the quadrate lobe, and here is the caudate lobe. This is the hepatic vein. Here's the hepatic porta hepatis, where we have the hepatic portal vein, hepatic artery proper, and common hepatic duct, all in this region. And then the structure right here is the gallbladder. So a lot of different in the gallbladder ducts as well, moving through. Gallbladder duct then wraps around and then comes out and joins with the pancreatic duct for producing bile. So gallbladder stores bile, produced in the liver, stored there, and then so it's made of smooth muscle that squeezes and goes out through these ducts and out through the pancreas and into the duodenum to help digest fats. So those are the main parts of the liver. There are some other parts here, like this is the falciform ligament, or the round ligament right here. And then that then goes up through and separates the 2 lobes and so forth. Let's clear that and move forward. So then I show that a little bit more, and then I jump over and show the different parts of the model. Well, the parts of that poster model. Like I said, it's not ideal, but it does get the job done. And the first thing I want to focus on here is this villus. And it works right here. So this is one villus that's found in our small intestines. So this structure that goes up through the middle here, that's the lacteal. A lacteal, so when we digest fats with bile surrounds them. And since then they're a lipid, they then get absorbed as what are called chylomicrons. These chylomicrons then get taken up in these lacteals, so you absorb fats into your lymph system. Everything else gets absorbed into all these cells right here. And we're going to see these cells, these structures, a little better on when I show the microscopic images, and they look pretty cool. So you can see all the cilia linings. They're all simple columnar epithelia that line this. And so this is a villus. Multiple ones are a villi. Then if we zoom in down here and blow up one of those cells has little ridges on top as well, and those are then microvilli, or one of them is a microvillus. So make sure you know the difference between a microvilli and a villi. They're different things. One is larger and the other one is a smaller one. And so this is the nucleus on top of one of those columnar cells. And the other things we have in here are the arteries and veins, so the capillary system that feeds up here. So when you absorb nutrients through one of these cells in here that come out the other side, it gets immediately picked up by the vascular system, unless it is a fat and enters through the lacteal there. Okay, so then that's what I wanted to focus on that. I'll go over a little bit more about that when we get to the microscope showing the different layers and so forth. Other things, so there's the appendix on this one. I showed the appendix on the other one. And there you can see the different portions. So we went over all this on the actual model, and this part, I kind of just wanted to play through this one now. To end it here, it's showing this version of the stomach. You have the fundus, greater, lesser curvature. That's showing part of the pancreas there, but we focused on that over there. So now what I want to do is switch over to the microscope. So let's switch over to, this is the Omax Microscope here, and this is actually a longitudinal section of the small intestine. So don't screw up a longitudinal section with a cross section. So typically if you would do a cross section, it'd be a nice round section with a hole in the middle where the lumen is. So you got to imagine that flipped to its side and you're cutting like this. So slightly different shape here, and that's why you don't see like a central lumen on this figure. So there would be one wall, and there would be the other wall. So if we zoom in there, we could see the different layers. And all these little structures running through the middle here are the different villi, or singular villus. But here, I want to focus on the different layers down here. So right now, we're at 40 times magnification. So going up to 100 times magnification now. I'm going to have to adjust the focus a little bit once it gets in here. So we can actually see some of the different layers now. And this is really cool. I might take an image here. So here, if I bring back my pen, right out here is the serosa layer. And then you gotta remember, this is a longitudinal cross section. So then right here is one muscular layer that's running longitudinally. And then right here, these are cells pointing out at you. So that's the circular muscle layer. There's the longitudinal layer. And then, so that's all the muscularis right there. And then we have the submucosa right here. So right there's the submucosa, and then right here's the mucosa layer where you have the actual absorption happening. Right here is, one villi. So right here is the lamina propria coming out through, the connective tissue coming out through. So the capillaries and the lacteal would be coming up through the center here and out. So it's really cool how that works. Let's see if we could take a nice little image of this real quick. Let's switch back to my cursor. So the way this works, I know you can't see everything being done on this program, but I pull up a little capture window here, and I want to go a little bit out of focus, about right there. So we capture the first image, and you'll slowly see me bring this back through focus now. And what this is taking a stacked image of this sample, and it makes the entire image focused rather than being out of focus in different regions. So I keep changing the fine focus just tiny little bits as I move through here. You'll see it's now getting out of focus as we get to the top portion. So right there looks good. I took 18 pictures right there, and then click through. I know you can't see these settings, but I can click through next. And now what it's doing right now is it's stacking all those images together. So a new panel is going to pop up with this final image, and we can see what it looks like and maybe zoom in and see some different structures and what we can see right here, because everything will be in focus together. So it's almost done, about 75%. It'll be ready in a second. And this is my new microscope here that I'm having fun playing with. So now everything's in focus. So it's super, super cool how that works. So here we can see these structures. You can even see these little cilia lining. So these little, along all these microvilli here. So here, these are all the enterocytes where absorption is taking place. Now you can't really see the different cells right there. That might actually look like a goblet cell right there. So a goblet cell would be a mucus secreting cell, and you can't identify like the lacteal or anything like that going up through there. But here, the deeper ones, these could be intestinal crypts coming down in here. So down here, one of these might be a paneth cell. And then, so you have your enteroendocrine cells as well, so releasing some hormones in here. And then if you're looking at the submucosal layer, now so right there, there's a circular cell. That one might be a MALT or a mucosa-associated lymphoid tissue, which would be found in the mucosa layer, and some are also found in the submucosa layer. In the submucosal layer, you can also find glandular cells as well, but I'm not seeing any of those in this one. Maybe there's one right there. And there also could be, so these darker granular cells, so the darker pigmented cell right there might be one of the granulocytes, such as like eosinophilic or basophilic cell. So you see those darker pigments in here too, or it could just be a cluster of stain. Depending on the stain, it's hard to differentiate different cell types, but we can do a decent job here. And this is actually a pretty cool image of this showing. You can see the different layers. There's the serous, and you can see these muscles running this direction. This is smooth muscle. So that's the longitudinal layer. Here's the circular layer. Here's the submucosal layer. And here we can see these different villi sticking up. So here's another cool one. There's a darker pigmented cell right there. So who knows what that one might be. I could process this image a little more and maybe take out a little bit structure, but I won't do that too much on this one. What I want to do now, just as food for thought, is go up to 400 times magnification and just see if we can make out anything else here. So there you can see like the actual cracks in the glass of the specimen of where it crystallized. So there, that actually might be a gland right there. So that could be, I'm not sure what part of the small intestine it could from. That could be something like a duodenal gland or just a mucus gland sitting down there. And here you could even see some of the stained structures. So all those pigments in there and the nuclei could be, so a lot of these cells are undergoing cell division too. So we could be seeing different rounds of cell division here as well, depending on the situation. Now, I'm not making out specific ones, but it's kind of cool. And some of these, so most of the dark pigmented stains are nuclei. So that's staining DNA, or it could even be staining RNA. Anything that's a nucleic acid is stained by that dye. So you can see individual chromosomes and whatnot in here. That's really, really cool. So I can also take an image here, but I won't. I might take one just to have for myself. So if we would change the focus here, let's see if we can find a special spot to look at. Here you can see this area down here a little better too. So if you change the focus, you can see how the fibers change. So those smooth cells are facing you right there because those are circular, whereas the ones right on the outside are longitudinal. And then here's the lamina propria going up in through the villus. Let's see, right here, it kind of, this is a little baby villus right here. Let's see if we can focus this one a little more. There are the different focus levels, and it looks like it's furry out here. And those are actually the microvilli, and some of them are also ciliated of these enterocytes, which are the absorptive cells in here. And you can see how they are a cuboidal layer in here as well, if you remember your tissues chapters in your epithelia. So no, I think this is really cool. I'm really happy I have this microscope at home now, and I could get to share some of the, ooh, that's actually a really good spot. You know what, before we end today, I do want to do a stacked image of this because it is looking like I see some cool structures in there. Then we get 2 stacked images, which will be really neat. So start capturing, and I know, again, you can't see the actual program that's running right now, but I am manually changing the focus as I move up through this sample, as you can see happening. Now this one, I won't have, last time I got 18 images. I won't get as many this time, because there you can start seeing the etchings on the glass. Okay, so I got 14. We'll put it back into focus for you. Then going through the program. So now it'll stack those images and go through them. So I know I'm only showing a small intestine. I believe I had pancreas, liver, and other tissue samples. And I hope to eventually make videos for all of those. It's just doing one at a time would have made this video way too long. So I'll probably make separate histology videos. Ooh, this one came out really cool. Okay, so it's done stacking. So if we're comparing, so this would be 100 times magnification right here. And then this one is 400 times magnification. You can see different structures down here. So these are different darker pigmented, maybe granular granulocytes, white blood cells. And then here you see the long enterocytes. Again, you can't make out like what's a Panus cell and whatnot from this image. But if there were, they'd be down there in these deeper intestinal crypts and so forth. But it is still cool to see these structures and try to figure out what everything is. Histology is always fun to do. It's just sometimes difficult to look at. The first time I looked at this image, I was really confused. And then I realized it was a longitudinal section. So here you can see some granulation in whatever cell that is right there. So maybe a mucus cell with the mucus granules or something else. I don't know. But yes, so we'll leave it here. Like I said, I'm glad I can bring these microscope images into these little laboratory videos now, but if that's all I have for today. If you have any questions, feel free to reach out to me. I hope this helped you see something a little bit more hands-on for digestive system models and so forth. But if you have any questions, definitely reach out to me. But with that, I hope you all have a great day, and bye-bye.